In Chapter 12, we're going to focus on what our DNA is actually used for. We know that it is a repository of information that is passed from one generation to the next, but what is the purpose of this information and how is it utilized? Well, the purpose is for us to be able to create particular proteins or gene products, use those recipes that are listed in the genome to create these things. But the first step in the production of these gene products is the transcription of the DNA. And transcription is a term that refers to utilizing some original copy to make a new copy or to create a new version. Okay? And that's what genetic transcription refers to. Outside of genetics, transcription simply refers to taking a transcript, right? You might read a transcript of someone's speech. It's someone writing down verbatim the words of a speech. Transcription does much the same thing in that we have cellular machinery that creates a faithful, accurate copy of the gene that we are focusing on. Okay? So the way it works is that the genes located in our DNA are accessed. One of those strands, not both, remember our DNA is double stranded, one of those strands serves as a template, and that's a term you remember from DNA replication, in which both strands are template strands for the new strand of DNA. But what we're creating here is not DNA. We are going to use one of these strands of the DNA as a template for transcription. Product of that process is a strand of RNA. This strand of RNA is going to be complementary to the DNA strand, the template DNA strand, and will then be translated using ribosomes that will then produce amino acids. So this is an overlook of the whole process. But recognize the chapter that we're dealing with right now, chapter 12, is just the first half. How do we go from DNA to RNA? The next chapter that we're going to review deals with taking the RNA, translating it, and producing polypeptides and proteins. Okay? So keep this overview in the back of your mind, but recognize we're only focusing on the production of RNA from a DNA template in this chapter. Now you'll remember that the genetic code is this linear form of bases, okay? And the code is copied or transcribed into RNA, and these genes in particular are copied into a single-stranded RNA molecule that we will call mRNA, the M standing for messenger. These messenger RNA molecules consist of a string of nucleotides, G's, A's, U's, and C's, guanine, adenine, cytosine, and uracil. And all of those nucleotides in sequence spell out instructions for the production of a protein. Okay. Now, the nucleotides are structured into what we call codons. Okay? These are 3 nucleotides read at a time consist of a codon. This has everything to do with the fact that our genetic code is, as you'll see a bullet point down, commaless, meaning there are no commas. There is no punctuation in our genetic code. The analogy I frequently use is that of a recipe for, say, cookies or brownies, right? That recipe is a list of instructions that you can read to produce cookies. You can read all of those letters on a piece of paper and interpret them, not simply because the letters are organized in a specific way, but also because there are spaces between words and there are periods and commas that separate different portions of the instructions. We don't have spaces, commas, or periods in our genetic code. So in order for the genetic code to be read consistently, the same way each time, it needs to be structured in a way that it can always be read the same way. And in order to do this, with only 4 nucleotide possibilities that can be arranged in many different ways, right? Our alphabet consists of a few dozen letters that can be organized in specific ways to convey specific information, okay? So you can read the word characteristics at the top of this page because some of these couple dozen letters in the English alphabet have been organized in a specific way, and there's a space between characteristics and the next word of on this page, okay? You can read that because the way the letters are organized, and you know that each unique word is separated by a space or punctuation from the next. When it's commaless, the genetic code relies on the consistency in the length of the quote-unquote words, these words being codons, okay? So the fact that our genetic machinery knows that every word, quote-unquote word, it is reading in our messenger RNA, in these instructions to produce a protein, every word is only 3 letters long, so it doesn't have to worry about where different words start and stop. Once it starts, it knows that it reads each word 3 letters at a time, one after the other. No spaces, no commas, no punctuation, okay? The code is also said to be degenerate, which we'll talk about here in a moment, and it contains start and stop points, okay? So when an RNA molecule is produced, it has this genetic information in 3-letter words that tells our cells how to make a specific protein. But as that recipe is being read, the cellular machinery, that being the ribosomes, need to know on what nucleotide they should start reading 3 letters at a time, and then also where to stop. And so for that, we have start and stop codons, okay? And these are used in translation, the topic of chapter 13, okay? And I've already talked about how the genetic code is commaless. The genetic code is also universal and non-overlapping, okay? And so these are important points that are discussed in detail at the beginning of your chapter. You'll find them on page 219 of your text. And I encourage you to review these and make sure you understand these characteristics. As we go through the rest of chapter 12, all of these should become quite familiar to you, okay? So the first thing I'm going to show you is what we call a codon chart or codon table. And this is a way in which we can visualize all of the possible 3-letter combinations or 3 nucleotide combinations that can be created by arranging G's, A's, U's, and C's, okay, in groups of 3. So if there are 4 letters arranged in 3-letter words, these are all of the possibilities. And you'll see that we'll start on the left side, work our way up to the top, and then the far right side. And so we start with, on the left, in the 4 main rows, what is the first letter of the codon? Is it a G, A, C, or U? Okay, so let's stay with the very bottom left codon, GUG, okay? Guanine, uracil, guanine. Having those 3 nucleotides in order in a molecule of messenger RNA is the word that, when translated, codes for VAL or valine, the amino acid valine, okay? So we can decode that codon, GUG, using this chart by knowing that it is in the bottom of the 4 left rows, G, because it starts with a G. The middle letter is U, so we go up to the top. The second codon position is U, and so that puts us in the first column. So now we are in the first row, so bottom row, and first column. And then the third nucleotide being G puts us in the very lowest row of that bottom row of G's. Okay, so GUG codes for valine. All right, now this is the simplest way, 3 letters, 3-letter words using 4 nucleotides that you can code for all of the unique amino acids that exist, okay, and that are utilized biologically. You could not do that with a 2-letter word using 4 nucleotides, okay? And if you used 4-letter words with 4 nucleotides, there would be way more options that are necessary in order to uniquely code for each of these amino acids, okay? So 2 things I want you to note here. The code is unambiguous, and it is also degenerate. Those are 2 things that were mentioned on the previous slide, and I want to show you, using this table, how both of those are true. The code is unambiguous because each of these 3-letter combinations, like GUG, there's only one amino acid, in this case valine, VAL, that GUG codes for. So when you see GUG in a codon, it codes for one and only one thing, unambiguously, all right? Now the genetic code is also degenerate in that GUG is not the only thing that codes for valine. GUA and GUC and GUU also code for that, all right? So let's take a look here at how what I've just talked about is utilized in translation. It's important you understand that, even though we're not covering translation in depth in this chapter. So here are 4 codons in the row in a messenger RNA molecule, okay? And you'll notice that the RNA is single-stranded. There's only one strand in translation. So a single-stranded RNA molecule is produced by transcription. It will then be translated. And this messenger RNA molecule has 4 codons in a row, AUG, GUG, UUG, and then AGC. You can look in the codon table or chart that's in your text. You can Google these. They're pretty easy to find, and see how that would be translated, okay? I want you to think of that codon chart that's in figure 12.7 of your text as basically the secret decoder for messenger RNA. And in translating these 4 codons, it would code for these 4 amino acids in this particular order, MET, VAL, LU, and SER, okay? So we have here in this codon chart a clear indication of exactly what each codon codes for in terms of an amino acid. You could decode any codon I give you, okay? And I also want you to pay attention to the fact that AUG towards the bottom left is highlighted in kind of a blue-gray box. And there are 3 up towards the top right, UAA, UAG, and UGA, that are stop codons, okay? And those 3 in particular do not code for an amino acid, as we will see in translation. These 2, the AUG, which codes for methionine, and these 3 stop codons, they are what I was telling you about on the earlier slide, that specify the beginning and the end, or the initiation and termination of translation. Think of AUG as the start codon for translation, and these other 3 as a stop codon. Any one of those 3 would stop translation. But everything in between codes for a specific amino acid that will be strung together during translation. So the text reviews how early studies utilized things like triplet binding assays that I'll show you in a moment to determine exactly what every codon codes for. We knew it was probably unambiguous. This was verified. But if the genetic code is unambiguous, how did we determine that UUU codes for phenylalanine? Okay, well, what biologists did is something like what is indicated here in this table. They used an isotope of phenylalanine in a protein, and they created artificial messenger RNAs that had only U's in it, or only A's, or only C's. And then they saw when they did that, and that messenger RNA was translated, what did the resulting string of amino acids contain? Did it contain lots of this radioactive or isotopic phenylalanine or not? And it turns out that if you just construct a long string of U's in a messenger RNA molecule and that is translated, what you get is something that is heavily labeled with this C14 phenylalanine. Okay, and so they determined that the codon UUU is likely coding unambiguously for phenylalanine. Okay, similarly, AAA would code only for lysine, and so on and so forth. Okay, so there were a number of studies that were used or undertaken to determine exactly what the genetic code is. It took a while to work out. Your text goes into a bit more detail than you really need to understand, but I want you to realize that it took a while for scientists to figure out which codons specified which amino acids. But once they did, it revealed the unambiguity and the degeneracy of the genetic code. So in using this triplet binding assay, they used ribosomes and tRNA molecules. All of this we are going to cover in the translation chapter. Okay? So, if you consider their use of ribosomes and artificially constructed messenger RNA molecules that consisted of just UGUG or UUG or UAUC, okay? Each of these presents some unique possible codons that could be created. So for example, in the first one, there are only two possible codons here, UGU and GUG. Using the triplet binding assay, they determined that those two codons coded for only two different amino acids. And through doing this iterative these iterative experiments that you see here, they were able to figure out what UUG coded for, what UGU codes for, and GUG and GUU, and so on and so forth, until we end up with the codon chart that you saw. Okay? Now, the degeneracy of the genetic code was identified because it became clear that multiple codons, like AAA or AAG, both code for lysine. Okay? And this is broadly true of the genetic code, as you see here. And this takes us to something called the wobble hypothesis. And this has everything to do with how translation takes place. Okay? But looking at this genetic code, I want you to understand that the wobble has everything to do with the differences in codons that code for the same amino acid. And for the most part, if you look here, what codon position is able to change and the codons still code for the same amino acid? What you will see is that for the most part, the third codon position can change. Within one of these primary 16 big rows and columns, so let's look at the bottom left where valine is. You can change the third codon position from GUU to GUC, to GUA, to GUG. This is that degeneracy or wobble, the ability of all four of these to code for the same amino acid has to do with the way in which translation takes place down the road, the way in which a tRNA is going to interact with a messenger RNA. Okay? So when we think about the use of these codons, I want you to understand the fact that different codons can code for the same thing, okay, because they differ in their third nucleotide in the codon, okay? Now, one of the possible exceptions to changing a third nucleotide position in a codon that would not have much effect is a change that would result in a stop codon, okay? So some of these changes could have serious effects, and we will talk about these down the road. But as you can see here, a mutation that changes CGA to CGG does not affect the amino acid that's coded for. But a mutation that changes UGU to UGA has a big effect in what that codon codes for, okay? So again, we will dig into this in our mutation chapter, okay? But I'm setting the stage now for you having some foundation for understanding what the point of the genetic code is and why transcription is necessary. And also, this will get you thinking about how mutations to the genetic code can cause issues in the production of proteins and result in diseases and other changes in organisms. Now, one of the bases of the genetic code that we talked about in one of the first slides here was that the genetic code is universal. And I have here an image that shows some common aquarium fish of different color. And the reason I use this image is because these are a laboratory created strain of zebrafishes, which are normally quite ugly, kind of silver and blackish. Now, these glowfish were created by a lab, and you can buy them at Walmart and PetSmart in town. And what's remarkable about glowfish is that the reason they have these unique colors, these reds and greens and oranges and even blues and purples, is not because zebrafish were mutated to produce these different colors. That would be far more difficult. What scientists did is they took the gene for the production of pinkish red color from a coral, and stuck it into the genome of a zebrafish. And then they grew that zebrafish from a single cell embryo and ended up with zebrafish that are expressing the gene product, this pigment, this pink pigment, of a gene that is from a completely different species, that is so evolutionarily distantly related that it's quite remarkable. And these fish can produce that coral pigment protein because the genetic code is universal. We could put that gene for coral pigmentation in a human, in a cat, in a rabbit, and make a pink cat or a pink rabbit, okay? Because the genes that differ between species are what create the differences between species, because we have different proteins. We produce different gene products, but we could produce each other's gene products because the recipes are all written in the same language. This is the universality of the genetic code, okay? So now that you have some background on the universality of the genetic code, the degeneracy of it, the way in which it is utilized in living organisms, now we're going to dig into this first step in the process of utilizing that information, and that is transcription. So the basics of transcription are we have the original DNA, right? Think of that as the original copy of the recipe. And what we need to do is make copies of it, transcripts of it, so that those transcripts can be sent out of the nucleus where the DNA is housed. And those copies, those RNA copies, can be translated, and we can actually express those genes. We can make that color in the fish, or we can make a protein in our bodies. We don't simply express genes, okay, translate them directly from the DNA for a couple of reasons. First of all, our DNA is housed in the nucleus. It's like the books you're not allowed to take out of the library, okay? This is the important reference material. If we want to express that locus and make lots of that gene product, we need to make copies, and those copies can leave the nucleus and be used in the expression of a gene, okay? We also generally need lots of copies of this gene in order to express or produce meaningful amounts of whatever this gene product is, whatever this protein is we're looking to make, okay? And since you only have in every cell 2 copies of every gene, one from mom and one from dad, that means all genes within your nucleus are at equal abundance, okay? But we don't need everything all the time in equal numbers. Some cells need some proteins sometimes. And so for that reason, we express them, we transcribe them, and then translate them when necessary, okay? So if we take these stretches of DNA that contain these genes, we are going to make a transcript. And that transcript is created through polymerization, just like DNA was produced during DNA replication. But we're using a different enzyme here. Instead of a DNA polymerase, we are using an RNA polymerase, okay? We definitely need these NTPs. You see those little orange things there in the top left. Remember, those are those nucleoside triphosphates. These are the raw building blocks of a nucleic acid. And we must, of course, have this double-stranded DNA molecule that has the gene. We have to copy the recipe, right? If you want to bring your mother's cookie recipe to campus with you so you can cook when you're in the dorm or in the residence halls, you make a copy of her recipe. And you don't just do it from memory. You look at her original copy and you copy that with great fidelity, minimizing errors because you don't want to screw up the cookies. So this RNA polymerase enzyme must access the information in the DNA directly so that it can be certain to make an accurate copy of that gene, okay? So the RNA polymerase, if it is to express a gene, let's say this gene, I always use this example, alcohol dehydrogenase. It's a protein my body needs whenever I have a glass of whiskey at the end of the day, let's say, okay? I need alcohol dehydrogenase, the protein, to break down this toxin in my body. And so when I need it, I express it. I have polymerases that will go to that gene and they will make RNA copies of it, right? So the RNA polymerase needs to have some direction as to where to begin this process. And so that is achieved by the interaction of this sigma factor that you see here at the top that is part of this polymerase enzyme complex. And the sigma factor identifies and attaches to a region right next to the gene we are interested in, called the promoter, okay? That promoter region, it gets a bit more complicated than this in eukaryotes, but the promoter region is there simply to say, here is what you're looking for, start transcription at this spot. The RNA polymerase enzyme comes in, opens up the double-stranded DNA, allowing the polymerase to begin synthesizing a new nascent RNA strand that is complementary to the template strand of the gene region in the DNA. You'll notice that there are two strands here labeled. One is the coding strand, the other is the template. So the template is the thing that is used as a template for transcription, and that means my RNA strand is going to be pretty much identical to the coding strand, with the exception of one having uracil, the other having thymine, right? So that RNA strand is complementary to the template. It is synthesized by the addition of nucleotide triphosphates to the 3' end. We make this in the very same direction that we make the DNA during DNA replication, okay? So this RNA is made in the 5' to 3' direction. Okay, once this process begins, the sigma factor disassociates. It's no longer needed. We know where the promoter was. We're already past it. We're making this RNA strand. This is transcription, okay? Now there are a couple of other elements other than just the promoter that we need to talk about. Some of these elements are portions of the DNA that are what I will call upstream of the actual gene region itself. These are regions that are associated with the gene, but they are not themselves transcribed. They are not copied in RNA by the polymerase. These things, like the TATA box, the CAT box, the GC box, these things are called cis-acting elements. And so by cis-acting, it means that they are elements that are right next to, physically connected to, very near the structural gene. And it is these things that are part of what I showed you as the promoter on the previous slide. These cis-acting elements occur in the region just upstream of the gene, and all of them together are part of what we generically call the promoter. Right at the end of the promoter is where transcription will begin. But it's this promoter region that interacts with other molecules, like that sigma factor I showed you on the previous slide. That sigma factor and other things, so there are many other transcription factors indicated by these purple things, or transcription binding proteins, this red molecule. All of these are other proteins that bind to portions of this promoter region, okay? Something upstream of the gene itself that binds to the DNA, and what they do is really facilitate the process of getting this RNA polymerase to actually sit down in the right place just at the beginning of the gene so that it can begin transcription. Now, you will remember that all of those portions of the DNA, the TATA box, the CAT box, all that stuff, what's labeled here as an upstream enhancer, all of those are called cis-acting elements. What these transcription factors and transcription binding proteins are called are trans-acting molecules, okay? So there are cis-acting elements and trans-acting molecules, okay? And they both interact with one another in order to facilitate the process of transcription, making an RNA copy of this gene, all right? So let's roll through the actual production of this RNA molecule. So the polymerase will slide along this double-stranded double helix of DNA, creating a new RNA strand that is complementary to the template strand. Remember, the sigma factor is disassociated. This RNA transcript gets longer and longer. And what that looks like, if we're looking at the double helix and the nucleotides that are hydrogen bound to one another, you will see that as the polymerase moves from left to right in this image, the double helix has to unwind. We have to break those hydrogen bonds so that the polymerase can read the template strand and attach complementary NTPs to the new RNA strand that's being made, okay? The coding strand here is ignored, and as the polymerase moves from left to right, the double helix gets wound back together over to the left after the polymerase passes by. So in transcription, we start at the promoter. We've got a few steps here. The first is the binding step. Then there's initiation, elongation, and termination. You can see these 4 numbered steps. Binding is simply the polymerase binds to the promoter site and starts unwinding the DNA. Initiation is the RNA synthesis starts. Now remember, unlike DNA replication, we don't need a primer, okay? We needed a primer because DNA polymerase can't do this. It can't just start from nothing. It can't start this new red strand from nothing. But RNA polymerase doesn't have that restriction. That's why we used that RNA polymerase called primase to create a primer in DNA replication. So here, transcription is initiated, and then the RNA transcript is elongated in the elongation step. And then when the polymerase reaches the termination signal, transcription terminates, okay? So transcription starts at the promoter and ends at the terminator. We're going to learn in translation about start and stop codons, and I already showed you those in the codon table. So remember that promoter and terminator sites are for transcription, start and stop codon are for translation. Okay, don't confuse the two. Now, what I want to remind you of here is that, remember, we only have 2 copies of every gene in your nucleus, okay? And we want to make lots of copies of it because I need lots of copies of alcohol dehydrogenase so that I can make a lot of it and digest this whiskey, okay? What you're seeing here down the middle is a strand of DNA, okay? A double-stranded DNA molecule that is being transcribed. And each of those little squiggly lines that goes to the top or the bottom is a different RNA transcript that is being produced through transcription as a RNA polymerase moves from left to right on this chromosome of mine, okay? And what you'll notice is there's dozens of them, okay? There are dozens of different RNA polymerases on my same gene lined up back to back to back to back to back to back, transcribing the exact same thing. What I'm going to end up with here in a few seconds is dozens and dozens of RNA copies of my alcohol dehydrogenase gene. All of those copies are going to leave the nucleus and go out there and be translated. So I can make lots and lots of alcohol dehydrogenase at the same time, okay? I don't want to make one alcohol dehydrogenase protein molecule at a time. I want to make hundreds of them at a time. So when we make this RNA copy of a gene, what it includes is a perfect RNA complement to our template strand of DNA, okay? It has a 5' and a 3' end, and it has a nucleotide for every nucleotide that was in that DNA stretch between the promoter site and the terminator, okay? Now, we have some work to do to this transcript before it can actually be used, okay? Now you'll notice there are 2 segments indicated here, I1 and I2. And what these represent are things called introns. Now, in that region of my DNA, in that gene, there are some portions of that DNA that are really important for the production of alcohol dehydrogenase. Some of those nucleotides, though, are not necessary. They're not actually part of the recipe. They're, let's just call them fluff or extra stuff, right? Maybe in your mom's chocolate chip cookie recipe, she made a note, that your brother loves to have butterscotch chips in the cookies. Well, you hate butterscotch chips. That's garbage to you. You're not going to write that down in your recipe. You're not going to use that information when you make it, when you make cookies, okay? These introns are the same thing. They are unnecessary information, and they cannot be there in this messenger RNA when it is used in translation, okay? So the first thing that we need to do to this DNA in transcription is get rid of those introns, and then we also have to protect this single-stranded RNA molecule, because in your cells, in my cells, single-stranded nucleic acids are very fragile. They get chewed up and destroyed, partly because, as you learned in the previous chapter, a lot of pathogens like viruses contain single-stranded nucleic acids, and we don't want that stuff in our cells, okay? So in order to protect our RNA, we need to put some caps on the end that protect it from being degraded from each end, okay? And so a methylguanosine cap is added to the 5' end. You can see that here, this little 7/mg on the 5' end. And on the 3' end, we've got a little bit of extra junk on there. We're going to cut off and add a bunch of A's to it, a poly A tail. Okay, so now this single-stranded molecule is much more stable. Our internal guardians of our cells will not come in and chew up this molecule from each end, because it is now buffered with a methylguanosine cap and a poly A tail. The final thing that needs to be done is getting rid of this extra information, these introns, okay? Because we cut those out, throw them away, what we end up with is an RNA molecule that contains only the exons, only the information we need to produce this protein. Now, the way I remember the difference between introns and exons is the introns stay in the nucleus. They do not get expressed. The exons get expressed, okay? So this is a mature messenger RNA molecule that could be translated, all right? So to summarize this, we've got a 5' to 3' coding strand of DNA here in green, and a complementary strand that is transcribed, okay, the template strand. So in transcribing the template strand, we produce what we call a pre-mRNA. This pre-mRNA doesn't have the methylguanosine cap, it doesn't have the poly A tail, and it still has exons in it, okay? I'm sorry, introns in it. And so we take this pre-mRNA, we add the 5' cap, we add the poly A tail to the 3' end, and we get rid of those introns. You can see how the introns that are in the template strand made it to the pre-mRNA, but they don't make it to the messenger RNA, the mature mRNA. Okay? And these introns can be pretty sizable. They get chopped out, and there can be a lot of stuff that gets chopped out. So previous editions of your text had this image and the next one, and what I'm showing you here doesn't make a whole lot of sense by itself, but when I tell you that what this is a strand of DNA that is lined up with and bonded to a complementary messenger RNA molecule of that same gene, okay? So there's a gene here, and it is lined up with the mature messenger RNA that results from transcription of this gene. And so this schematic makes it more clear. The DNA is pink, the messenger RNA is orange, and you'll see that there are big chunks of the pink DNA strand that don't line up with anything in the messenger RNA. Where the messenger RNA and the DNA are lined up right next to each other, they have the same complementary nucleotides. The template strand and the mRNA line up perfectly. If I show you the gene and show you how long each of the introns in green and exons in orange are, you can see that the big loop A corresponds to that 1150 base intron, okay, in the gene. And then that loop B, that's super tiny, corresponds to a 246 nucleotide intron. That loop C corresponds to a 576 base intron, okay? So you can see all of these introns. The loops are proportional to the length of the intron. The longer the intron, the bigger the loop, because the DNA and the RNA don't match, because that chunk of DNA has been cut out, or that chunk of RNA has been cut out of the RNA, messenger RNA molecule, when the introns were removed. So transcription is not terribly complicated. It's very much like DNA replication. Go back and look at it. We were making that RNA in the 5' to 3' direction. We were reading the template strand in the 3' to 5' direction, the same way we read DNA when we replicate DNA, okay? We then remove the introns, we add a poly A tail, we add a 5' methylguanosine cap, and that's it. That's transcription. We're making an RNA molecule, okay? Now, most of these molecules of RNA in this chapter were listed as being mRNA. Now, there are a bunch of different types of RNAs, and we are going to talk about many different RNA types, particularly in the next chapter when we cover translation. What I want you to recognize is that every RNA molecule that is made within our cells is made through transcription, okay? Now, not all of them are transcribing genes necessarily, and many of them are not then translated. But every RNA molecule that's made in our cells is made through the process of transcription. RNA polymerase goes to a portion of DNA and makes an RNA copy of it. That's transcription. Some of those transcripts become mRNA molecules, some of them become other things. It depends on what was copied and what they are to become. Okay, now the last thing I'd like to touch on in this chapter is the principle of alternative splicing. Okay. Now, I talked to you about how introns are cut out and thrown away, and the exons are retained, and that's what's shown here in A. Okay, the pre-RNA had a bunch of exons, a blue, a red, a green, and an orange, and they were separated by some introns, the black lines. Those black lines get cut out, and all the exons get glued together, just like that mRNA at the top right. A principle that we are going to cover in more depth in, I believe, chapter 16, alternative splicing, is one in which sometimes we don't always cut out the introns and not the exons, okay? We can, through alternative splicing, skip certain exons. So look at the next two examples of the messenger RNA. One of them is missing the green exon. That is possible if we just remove all the introns and remove the green exon, okay? Sometimes we might remove or include portions of an exon, as you see here in the alternative 5' splice site or 3' splice site, okay? Sometimes introns can be retained, and sometimes there can be mutually exclusive exons. Basically, what I'm showing you here is that the same gene can be transcribed, but then the processing of the messenger RNA can proceed in a number of different ways that give us different mRNAs. We'll cover this in more detail later, but this is principally why your body has only about 20,000 genes, but you produce about 100 different thousand things with that. So you can do that if you include or exclude subsections of the recipe, if you will, and produce alternative mRNA forms from the transcription process, depending on the needs of your body, okay? So we'll cover that principle in a great deal more detail later, but it's important to introduce it at this point.